guys, today we are from group 2. We will presentation about carbohydrate metabolism or metabolisme karbohidrat. Let's get started. First of all, I will explain about definition carbohydrate. Carbohydrate is nutrients that the body needs in large quantities and can produce energy because it contains calories. Artinya, karbohidrat adalah nutrisi yang dibutuhkan tubuh dalam jumlah yang besar dan dapat memproduksi energi karena di dalamnya terkandung kalori. Next, I will discuss about the function of carbohydrate. Structural basis of many organisms. For example, cellulose of plants, exoskeleton of insect. Main source of energy in the body. Storage form of energy. Excess carbohydrate is converted to fat. Artinya kelebihan karbohidrat itu akan menyebabkan kegemukan. Glycoproteins and glycolipids are components of cell membranes and receptors.
Monosaccharides, known as exoses, consist of three types, namely, glucose is known as vegetable sugar found in vegetables, fruits, syrup, and corn. Second is fructose, is the sweetest sugar, and lastly is galactose. Galactose is the result of lactose digestion, which is not found in nature. Disaccharides, consisting of three types, namely, first is sucrose or sucrose referred to as cane sugar, 99% granulate sugar consisting of sucrose and sucrose found in fruits, vegetables, and honey. Second and third is maltose and lactose found in the intestines, and lastly is trehalose, also known as mushroom sugar found in mushroom.
Next is the simple type of oligosaccharide found in grains and legumes. The type of oligosaccharide cannot be broken down by digestive enzyme. Next, we move on to the simple type of polysaccharide. There are four. The first one is starch. The function of starch is as energy reserve for plant. The second is glycogen. Glycogen is a form of food reserved in the body. The third is cellulose, found in plant cell walls. And the last one is chitin, can be found in fungal cell walls and insect shell.
Carbohydrate digestion begins in the mouth. The salivary glands in the mouth secrete saliva, which help to moisten the food. The food is then chewed while the salivary glands also release the enzyme salivary amylase, which begin the process of breaking down the polysaccharides in the carbohydrate food. After the carbohydrate food is chewed into smaller pieces and mixed with the salivary amylase and other salivary juices, it is swallowed and passed through the esophagus. The mixture enters the stomach where it is known as chyme. There is no further digestion of chyme, as the stomach produces acid which destroy bacteria in the food and stop the action of the salivary amylase. After being in the stomach, the chyme enters the beginning portion of the small intestine or the duodenum. In response to chyme being in the duodenum, the pancreas releases the enzyme pancreatic amylase, which breaks the polysaccharide down into a disaccharide, a chain of only two sugars linked together. The small intestine then produces enzyme called lactase, sucrase, and maltase, which break down the disaccharide into monosaccharides. The monosaccharides are single sugar that are then absorbed in the small intestine. Carbohydrates that were not digested and absorbed by the small intestine reach the colon, where they are partly broken down by intestinal bacteria. Fiber, which cannot be digested like other carbohydrates, is excreted with feces or partly digested by the intestinal bacteria.
Next, we move to carbohydrate metabolism.
Carbohydrate metabolism is a fundamental biochemical process that ensures a constant supply of energy to living cells, which can be broken down via glycolysis, enter into the Krebs cycle and oxidative phosphorylation to generate ATP.
Humans get carbohydrates from their food. Food that contains carbohydrates such as rice, bread, fruit, and others will pass through the digestive process. Then all substances needed by the body will be absorbed, thus producing the energy needed by humans to carry out daily activities.
The process of glycolysis, the process in which a glucose molecule is broken down into two molecules of pyruvate. The end products of the reaction include 2 pyruvate, 2 ATP, and 2 NADH molecule. The glycolysis pathway occurs 10 following steps. Step 1. A phosphate group is added to glucose in the cell cytoplasm by the action of enzyme hexokinase. Stage 2 or step 2, glucose 6-phosphate is isomerized into fructose 6-phosphate by the enzyme phosphoglucomutase. Step 3, the other ATP molecule transfers a phosphate group to fructose 6-phosphate and converts it into fructose 1,6-bisphosphate by the action of the enzyme phosphofructokinase. Step 4, the enzyme aldolase converts fructose 1,6-bisphosphate into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate, which are isomers of each other. Step 5, triose phosphate isomerase converts dihydroacetone phosphate into glyceraldehyde 3-phosphate, which is the substrate in the successive step of glycolysis. Step 6, this step undergoes two reactions. 1. The enzyme glyceraldehyde 3-phosphate dehydrogenase transfer one hydrogen molecule from glyceraldehyde phosphate to nicotinamide adenine dinucleotide to form NADH plus H plus. Glyceraldehyde 2 glyceraldehyde 3-phosphate dehydrogenase adds a phosphate to the oxidized glyceraldehyde phosphate to form 1,3-bisphosphoglycerate. Step 7, phosphate is transferred from 1,3-bisphosphoglycerate to ADP to form ATP with the help of phosphoglycerokinase. Those two molecules of phosphoglycerate and ATP are obtained at the end of this reaction. Step 8, the phosphate of both the phosphoglycerate molecules is relocated from the third to the second carbon to yield two molecules of 2-phosphoglycerate by the enzyme phosphoglyceromutase. Step 9, the enzyme enolase removes a water molecule from 2-phosphoglycerate to form phosphoenol pyruvate. Step 10 or the last step is a phosphate from phosphoenol pyruvate is transferred to ADP to form pyruvate and ATP by the action of pyruvate kinase. two molecules of pyruvate and ATP are obtained as the end of products.
Glycogen is a polysaccharide homopolymer that is stored in tissue and stored as carbohydrates. During hydrolysis, glycogen is converted to glucose. Glycogenesis is the process of forming glycogen from glucose. Glycogen is synthesized according to energy requirements. If there is sufficient insulin in the body, excesses glucose will not be used and will only be stored in the form of glycogen.
And this is the glycogenesis pathway or steps of glycogenesis. First, glucose phosphorylation. In the initial phase, glucose is phosphorylated into glucose 6-phosphate, a usual reaction in glycolysis. It is catalyzed by glucokinase or liver and hexokinase muscle. Second, conversion of glucose 6-phosphate to glucose 1-phosphate. An enzyme phosphoglucomatase will catalyze the conversion of glucose 6-phosphate is converted to glucose 1-phosphate. Third, UTP or uridine 3-phosphate attacks to glucose 1-phosphate. The third step focuses on the reaction of glucose 1-phosphate to UTP terabyte forming active nucleotide UDP glucose or uridine diphosphate glucose. The one responsible for such reaction is the enzyme UDP-GLC pyrophosphorylase. 4. UDP-GLC attaches to glycogen primer. A small fragment of already existing glycogen will serve as a primer in order to stimulate the synthesis of glycogen. The glucose from UDP-GLC will be accepted by glycogenin. The initial glucose unit is attached to the hydroxyl group of tyrosine of glycogenin. The first molecule of glucose is transferred to glycogenin, which will then take up for glucose residues, forming a fragment of primer. It will be the one to accept all glucose molecules. 5. Glycogen synthase synthesizes glycogen. Glycogen synthase transfers glucose from UDP-GLC to glycogen, non-reducing end, forming alpha-1 for linkages. The same enzyme catalyzes the synthesis of the unbranched molecule with alpha-1 for glycosidic linkages. 6. The formation of glycogen branches. The final step is the formation of glycogen branches caused by the effect of branching enzyme, which transfer a small fragment of about 5 to 8 residues of glucose from the non-reducing end of the glycogen chain to another glucose residue linked by alpha-1 6 bond. This action causes the formation of a new non-reducing end. The final result is the elongation and branching out of the glycogen chain. Next is glycogenolysis. Glycogenolysis is the biochemical pathway in which glycogen breaks down into glucose 1-phosphate and glucose. The reaction takes place in the hepatocytes and the myocytes. The process is under the regulation of two key enzymes, are phosphorylase kinase and glycogen phosphorylase.
The hexose monophosphate chain, also known as the pentose phosphate pathway, is a unique pathway used to create products essential in the body for many reasons. The HMP chain is an alternative pathway to glycolysis and is used to produce ribose 5-phosphate and nicotinamide adenine dinucleotide phosphate NADPH. This pathway occurs in the oxidative and non-oxidative phases, each comprising a series of reactions. The HMP chain also has significance in the medical world, as enzyme or cofactor deficiencies can have potentially fatal implications on the affected patient. Glucose 6-phosphate in the presence of NADP and the enzyme glucose 6-phosphate dehydrogenase forms 6-phosphogluconodiolactone. The first molecule of NADPH is produced in this step. The 6-phosphogluconodiolactone is unstable and the ester spontaneously hydrolyzes to 6-phosphogluconate. The enzyme that catalyzes the reaction is lactanase. 6-phosphogluconate further undergoes dehydrogenation and decarboxylation by 6-phosphogluconate dehydrogenase to form the ketopentose, the ribulose 5-phosphate. This reaction generates the second molecule of NADPH. The enzyme phosphopentose isomerase converts ribulose 5-phosphate to its aldose isomer, the ribose 5-phosphate. In some tissues, the hexose phosphate pathway ends at this point, and its overall equation is the net result is the production of NADPH, a reductant for biosynthetic reaction, and ribose 5-phosphate, a precursor for nucleotides.
Next is gluconeogenesis, the formation of new glucose molecules in the body as opposed to glucose that is broken down from the long storage molecule glycogen. Gluconeogenesis is the opposite process of glycolysis. It takes place mostly in the liver, though it can also happen in smaller amounts in the kidney and small intestine.
So now I will explain you the process of gluconeogenesis. Gluconeogenesis begins in either the mitochondria or cytoplasms of the liver or kidney. First, two pyruvate molecules are carboxylated to form oxaloacetate. One ATP or energy molecule is needed for this. Oxaloacetate is reduced to malate by NADH so that it can be transported out of the mitochondria. Malate is oxidized back to oxaloacetate once it is out of the mitochondria. Then, oxaloacetate forms phosphonopyruvate using the enzyme PAPCK. Then, phosphonopyruvate is changed to fructose 1.6-biphosphate and then to fructose 6-phosphate. ATP is also used during this process, which is essentially glycolysis in reverse. Fructose 6-phosphate becomes glucose 6-phosphate with the enzyme phosphoglucoisomerase. Glucose is formed from glucose 6-phosphate in the cell endoplasmic reticulum via the enzyme glucose 6-phosphate. To form glucose, a phosphate group is removed, and glucose 6-phosphate and ATP become glucose and ADP.
The conclusion of this presentation is carbohydrates are nutrients that the body needs in large quantities and can produce energy because it contains calories, whereas carbohydrate metabolism is a fundamental biochemical process that ensures a constant supply of energy to living cells. In the process, there are 5 types including glycolysis, glycogenesis, glycogenolysis, HMP shunt, and gluconeogenesis, all of which have the same goals. They are energy production, energy storage, building macromolecules, sparing protein, and assisting in lipid metabolism. Okay, that's it for our presentation about carbohydrate metabolism, and we would like to say thank you for your time and attention.